The coral?
Yeah.
Roger.
Yeah.
All right. You can zoom out as I land.
What kind of coral is this one?
I'm gonna get a better zoom first before I say. Okay, there's two things here actually. I want to look at both of those things.
Yeah. There's something that's stuck to the rock.
Yeah,
so while we do this, I'm gonna pause our sampling. Okay. So if you can pause 8 and 9, if you can record that, please.
Just to pause, just to confirm.
A little bit. Yeah, I'll say just a pause.
Do you want to see your porch as you land?
Yeah, I'd love that. I'm just going to park it right in front of the porch there and try to land it. It looks like a small
I'm not sure if this is a coral yet,
you can
but
iris up video,
I'm pretty sure that it is, but I'm not 100%.
Roger.
Yeah, perfect.
But if it is a coral, it's one we don't see often. I see there's a feeding trace here. I don't know if you see that. You see how it's the rock is lighter? So that's what I think. This thing is a deposit feeder, and this coral is around it. But this is all free of sediment.
Okay.
See how it's different color?
Yeah, let's zoom in. I'm sitting on the ground now. Let's see what we can do here. What do we think this is? Not a good angle, really, unfortunately.
Probably tilt over a little bit. Yeah. There you go.
Pretty small.
Okay, yeah. So we have here.
What is this? Oh, is it like a parasitic? What am I looking at there?
So
kind of get
that's
parasite
really
energy.
cool.
So this thing on the left is a bryozoan, not a coral. But this thing on the right is very interesting.
Should we scrape it off?
I think we're going to have to, but it's going to be extremely fragile.
Fragile. So like suction sampler and like kind of get it to come in?
Yeah, you got to coax it. Probably if you come in from this side, it'd be better off because, yeah.
Do we have any vague guesses for what this is, or am I just putting
So
unknown?
it's either a limpet or a Multipuffer. I'm leaning towards limpet, but I just don't have good three-dimensional view of it. Yeah. This is actually more interesting than that. Sorry for the bryozoan people right now. all one of our bryozoan viewers.
Okay. Yeah, let's zoom out. Let's look at the suction hose and then we'll grab that.
It may come easily, it may not come easily. It's going to be really tough to tell.
Thing on the ground.
Do we know what it is? Or?
No.
Let's call it limpet for now.
Okay.
And we actually do have a request for limpets.
Should I go to?
Are we thinking to put it in a suction jar?
Yeah.
Cool.
All right, looks like I'm on 3. Is 3 vacant?
Yeah, 3, 6, and 7 are all
3
open.
is vacant, okay.
Which one has the squat lobster in it?
The squat lobster is in 1.
Okay, perfect. We'll just make sure we don't
linger
on that one.
There's a fish in one of them too.
Really?
Yeah. There's like
the fish is in 5.
5, yeah. So when after 3, I go to 4. 4 should be good. All right. Do you see the angle he said to come from?
Limpets are mollusc.
So I think if you can bring the hose around, like from this way.
Okay, we'll see if I can get the suction. We'll see. Yeah.
All right.
You might have to move.
Tell me what you need.
Yeah, we'll see. You might have to move, but sorry, you can see I'm not used to running with the blue button on.
Copy.
The reason I say to come in from that direction is typically gastropods don't like being pushed from the back.
Yeah.
And so if you push them from the back, they often release easier than trying to scoop them from the front.
Steve's done this before.
That's right. The limpets are a type of mollusk.
There's first rodeo.
Limpid rodeo.
Marine snail in the class Gastropoda.
All right. There we go.
Yeah, I'm leaning towards limpet on this, but I just the shell is so translucent, it's hard to get some contrast. But you can see the internal anatomy quite well.
Yeah. I'm just gonna get a view of the suction hose because I'm a little bit worried we're pulling that really far.
Yeah, you can see it in the in this camera.
Yeah, how's that
Looks
bend
fine.
angle look fine?
Looks right, yeah.
Okay.
You're basically there.
All right, is this...
Oh, you're deep, you're deep. You gotta come up.
Okay.
Up. Yeah. You got me suck? Here we go.
Yeah, do it.
All right. Okay. We're inhaling on dry 3. Oh.
Oh, don't.
There you go.
Easy. Steady. Okay.
There we
Okay.
go.
Okay. And I'm gonna kill it now. Okay.
All right.
I killed it.
Super.
And I'm gonna go to 4 now.
Good job.
Is it still in there somewhere?
It's
It's not gonna swim, so. Huh.
Where are
Maybe
you,
it's against the
friend?
filter. There it is.
there it is.
Awesome. Eagle has landed.
Okay.
Yep.
All right,
Nice.
it's in 3.
Yep.
Nav, could we get a pin drop for 050?
I'm gonna zoom us out video. We'll stow this
Thank you very much.
This schnozzle.
Schnozzle.
And then I think we're just going to go right into sampling that bryozoan next to it too.
Oh, okay.
Roger
So we're
that.
taking
With
the
that's a grab and probably front toolbox.
Roger.
Copy box.
I gotta depth perception this a little bit.
Yeah.
All right, bonk. Sorry. And,
It's pretty small.
I got an index.
Yeah.
Let me get a sample.
Are we
in
taking the
situ.
whole, like we're taking
Yeah,
the
it's gonna pop off the rock, the whole thing
Okay.
easily. They don't
Perfect.
strongly adhere.
It's a
Yeah.
small sample, so.
Okay.
Okay. I think I'm gonna get nice and I'm just getting, yeah.
Very nice.
I think.
It's pretty small, so you're gonna have to get as close to the rock as you can get. Yeah.
So this is gonna be really brittle, right?
Yeah. Look.
Yeah, let's say yeah.
Like, am I?
You just gotta go for it. Yeah.
Okay. Should I approach it from a different angle?
No, I think you got. I think
Okay.
I think that's great. I think just
We're
close the
gonna
jaw and whatever you get,
do
you
it.
get.
Oh.
Yeah,
Apologies,
we'll take it.
everybody.
You got it.
We could vacuum up the pieces
Yeah,
too.
I think we should do that.
Or what do you want
I
to
think
do?
just yeah.
Well,
don't
I'm
know
glad
if I
the
got
sampler's
any-
off because otherwise we'd be getting an awful lot of bryozoan DNA.
Yeah, I think it's just stow what you have in the box.
Okay.
I think yeah,
Okay,
it'll
I'm
take too
ready
long
to
otherwise.
Forward box open when you are.
Preferably the Omega bio box because we already have a coral in there.
Yeah, Roger.
I didn't know they were gonna be that.
All right, it's all the way
Mixing
open.
time.
Is there anything I can do to help you here? You want me to retract my camera?
Yes, please
Alright,
retract
alright,
camera. Yeah,
alright.
sorry. I should have checked before I started.
All right, it's
Popping in here.
retracted now.
And I believe
Omega. I like your symbols.
I am in here.
These are two separate
Alright.
samples, but since
Yes.
I don't know if you should do two separate pins. They're the same
There you go.
location
There you go.
either way.
There's some little 4 pieces went in, 5 pieces are in.
Yeah.
There you go.
That's
You got
enough.
some, Steve?
Yeah. Bryozoans are
Alright.
colonial, so if we have a small piece,
I'm
we have
clear.
enough.
There's something on the ground past the arm.
Yes.
Oh yeah, the enemy.
Yeah.
What was the question about 2 separate samples?
All right.
Cool.
Nav was asking me
Okay, yeah.
if those two were two.
Okay.
Okay, getting the arm out of the way. Here.
Are these 150s or 151?
Yeah, sure. All right. Yeah. So were we expecting that sample to be as brittle as it
No,
was?
I
Okay.
was not expecting that actually, but yeah, I think either way, suction or otherwise, I don't think it would have been much better. So I think we did the best we could. Super. We are doing really well this watch. And it's only halfway over. Think of how much more fun we can have.
Thank you. Say again? Yes, that's right.
Yeah, this watch has been amazing. So much stuff compared to last watch.
Oh, jar 4. Okay. Perfect. Roger.
I have a black coral in Slurp Jar 4.
You have a black coral in Slurp jar 4. Okay.
I think it might be up in the
Okay.
mesh.
I'm gonna. Yeah.
I
Okay,
did
I'm
see
just
them
gonna
collect that earlier.
Blast through this.
Are we slurping, suctioning
Let's
anything?
go. Ooh, come on. There we go. Yeah, let's do it. Yeah.
You can just pass it to them whenever they get a chance. I know they're busy.
So I think we are going to say that those last two samples, instead of pausing, I'm just going to wait a minute and start a fresh sample. So consider them to be finished if you need to update the data log.
Okay. Could I get the volumes?
Yeah. Oh, give me a second here. These were pumps 8 and 9, right?
Yep.
Okay. Pump 8 is...
Oh, interesting. Let's take a look.
Yeah, pass them around.
206-625.
Not my stickers, just the candy.
625.
Yes.
Okay. And then pump 9.
192-343.
192-343. Thank you very much.
Yep. Can you read back to me what I told you for pump 7?
The final volume?
Yes.
1 moment.
Do you have any port, Jen?
Pump 7, I had 194-743.
Yes, that's correct. Okay,
Great.
Thank you.
Thank you. And then I'm getting ready to start 8 and 9, correct? Or no? Sorry.
Yeah, sorry. It's 10 and 11.
Yeah.
And
10.
are we moving now? So, hang on a second here. Okay, starting 10 and 11 now.
All right. I don't know.
Could I get a pin nav for 052 and 053? Yeah.
So this is a stalked crynoid here.
You want me to zoom on it?
Yeah, if you quick zoom.
Just to confirm, this is pumps 10 and 11?
Correct, yes.
Thank you.
Oh, sorry. Everybody, bear with me. I'm relearning sticks.
We have faith in you, Ra. You got it.
Thanks. It's been a while. All right.
Yes.
There
It's
we
a
go.
multipuffer.
So I mentioned earlier the two groups of crinoids, the stalked crinoids called Culeolus and then the comatulids, feather stars. This is the stalked group.
Right.
They're a totally separate evolutionary lineage than the other feather stars.
Oh, wow.
And they're ancient. They actually are
you can
very
go
well
up.
represented in the fossil record.
Amazing.
This one has, see the fuzzies on the stalk are
up.
probably hydroids. So those are extremely common to grow on the stalks of other things. That's good. That's a good view of that. Where are we at now? 24 or 40 meters. Making some progress.
I'm pretty stretched out, aren't I? Yeah, sorry.
Where are we at again?
I'll just go sideways for a bit,
24-41.
The last waypoint is expected to be around 2180 meters, so we've got a bit to go.
Oh my gosh, we have so many people from all around the world watching.
That's cool.
Super cool. We've got Brazil, Germany, Guam, Sweden, Thailand, Malaysia, Australia, New Zealand, Marshall Islands, Canada, and the US. Super cool to have all these visitors watching along.
All right. I don't have the eye for like what's interesting to look at, honestly. So if anybody sees anything, just tell us straight.
Gotcha.
It's a bit of a Where's Waldo.
Yeah, I'm much more like operationally focused, but
It's definitely colder over here.
is it? Okay, because
Yeah.
I was freezing and I see it.
The
Yeah.
pilot seat's a little bit warmer.
You didn't bring a hoodie.
I know, it's pretty... Oh, Dan, give me a
Oh,
blanket.
Oh.
shit.
This is the sweetest
Sharing is
thing.
caring.
He's got his blankie.
It's so nice. Okay, I'm totally fine now. Aw.
Dan, is your guys' seat pretty cold?
He's
Yeah,
got
you
a,
got the, yeah,
he's
yeah.
got like gloves on, basically, and polar fleece on. Ooh, a sea slug.
Sea slug. Is it sea slug or is it a cucumber?
Cucumber.
Okay.
Yeah.
Are those interchangeable terms or no?
They are not. They are two different
I'm sorry.
groups. Yeah.
Sorry, I wasn't trying to blast you.
Yeah, you know, that's fine.
I was
Just
curious too.
immediately-
Do you want to snap on that, or are we
that
good?
one we've
Do you
seen?
want to snap zoom on
Yeah.
that?
We've seen it.
That really pretty
Alright.
pink one we saw earlier.
Oh.
Seen one cucumber, seen them all.
The cucumber.
They're pretty common,
Access.
but
What about this
occasionally
little-
there are small
Such
differences.
a way.
Coral thing.
And you were saying they can get pretty big, right?
Let's see.
They-
They can get solid,
Close to
foot,
30,
foot and a
30
half?
centimeters long.
Oh,
yeah,
wow.
something like that. Maybe bigger. But the problem is
Yeah.
they're mostly water, so
Dan, you want to snap zoom on this?
appearance
or
may change when you
zoom
bring
on
it up.
this? I don't know what this is.
Gotcha.
yeah, let's take a quick look at that.
It's like a tiny micro
It's
coral.
very small, but
They
yeah,
asked
this
for
is a small golden coral or small Chrysogorgia coral.
So what's up? Oh, sorry.
We'll keep looking because we've seen bigger
Okay.
ones of these,
Roger.
but
Okay.
we'll just make a note of it.
Zooming out.
Okay. Sounds
Sorry, what was
good.
that we're making a note of?
Chrysogorgia coral. I'll put it in sea log.
Awesome.
Zoom outs can be a bit faster for some of these. I'm gonna struggle a bit more to center my zooms up than Levi was, so sometimes maybe give me a bit more time, but yeah, cool. Some of this rock. Little white speck.
Yeah, these rocks are looking pretty round and crusty.
Yeah. Val would not
I'm
like them.
waiting for the cube on the rocks, but I just think the first rock was just so perfect that
The one earlier today?
yeah, the first
Oh
rock
yeah, it
was
great.
that was like a brick. Like
Yeah, it was like a perfect square almost.
somebody
I'm gonna zoom in on this white.
Someone chiseled it away.
yeah,
Just trying to see if it's a thing or is that just
It's like
What is
remnants
that?
of an old holdfast.
it's an
maybe?
old
Yeah.
barnacle shell.
Gotcha.
Oh, alright, zoom out. Yeah, that's a great speed for zooms out. Let's see, I think there's a little shrimp over here. I'm gonna zoom on the shrimp.
Oh yeah. Hey little friend. So what is
Yeah,
he?
so this
Just
is,
under 10 centimeters?
This
No.
is
Yeah.
this is a shrimp
Yeah.
called the Metacarcinus. They're long-legged shrimp. You can see they're usually. Sometimes you'll see them feeding on the sediments. Sometimes they're. You see them swimming and filtering.
Alright. You can zoom out.
Yeah, and for those of you watching, the way we're able to tell the size, those two little green lasers are 10 centimeters apart. So they let us know a little bit about how big these organisms are.
And that's just another crynoid that we've been seeing.
Yep.
Alright,
Yeah.
I'm gonna get back in there.
Crynoid might be the most abundant thing on this dive.
Back in our
Probably
hay.
one or two species we've seen so far. They're often collected, but yeah, crynoids are a tough group. The field has lost some really important taxonomic experts in recent years to retirement and such.
I feel like as soon as I gave you the sticks, there's nothing to look at.
That's okay. Also waiting for Atlanta, so.
Oh.
Hurry up, Levi.
Hey, you can't rush.
Can't
rush
perfection?
progress. You can't rush science.
Yeah.
Quality is a race with no finish line.
Ship is at the end of this movement.
Do we want to do another one?
Did that one just move? That ship
That
move just completed?
finished?
Maybe
Yeah.
we'll wait just a minute, I think.
Okay.
Let's see. It doesn't look very steep though.
Yeah.
If you wanted to know, we could, but we haven't felt that move quite yet. So
Yeah, roger.
I'm about to pull up some quotes.
Some quotes?
Some Matt Damon, like, on
Matt Damon.
YouTube.
Do it.
I'm trying to think of, like, Interstellar, like Cooper.
Okay, teach me about bias.
I'm trying to
See
think
bias.
of good quotes. All right, so then right now it's set to 25. So just root is to like 20. Roll it till that says 20. Yeah, the right one, yeah. Or that's strike gain. So, bring that back up to 100, strike gain. Peg that to 100. Like, okay, and then Z bias is the left one, and make that like 20. And then just fly around with that and see how that feels. Yeah. Was that joy gain that low for the, like, my whole shift?
Yeah.
Oh, that explains
That
a lot.
explains.
All
I
right.
mean, sometimes, actually, I'm going to turn it to 70 because I kind of like that, but
Okay.
just so that
Yeah.
I find that, like, nice when you're doing your zooms. You're not like,
Mm-hmm.
oh.
Yeah.
But
You can, yeah.
that's funny.
There, it's a stalked something or other. See that in your wide angle? I'd go to that next.
Oh, yeah.
See that
right.
stick?
Yeah.
Yeah.
I have to do a shout out real quick from someone very special to me. Yeah. Casey back in Fayetteville.
Perfect.
So glad you're watching.
Let's see if I can. Yeah, right. That makes sense why I
Yeah,
was
Fayetteville.
Just to the left. See the stalk?
Yeah, I see it.
Yeah. Steve's gonna wait for the reveal. Boom.
Whoop.
No one has ever seen this particular individual before.
It looks exactly like the last one we saw.
What's he got on the top, though? Oh, so is this a
Dead sponge.
dead one?
Yeah,
Okay.
this one's not topped with the flytrap anemone. But yeah, these all look like the bolosoma stalks we've been
Yeah,
seeing.
if you zoom in, I'll kind of pan up it.
Mm-hmm.
Those
You need
little
me to control
brittle
your
stars just look
camera
like little
tilt,
I'm
bracelets
just gonna
wrapped
try
around
to
it.
float it. I just need to
Float
get
it?
used to like my controls again. And just doing it smoothly, but yeah, I'm gonna like it's nice. So are these brittle stars getting, are they just
Quotes
getting
are coming in
like, nutrients?
for
Yeah,
Matt
they
from
may
it,
be grazing on some of the stuff that lives on the
Okay.
sponge stalk. Yeah.
or- Yeah.
Yeah.
Neat. Cool. Alright.
But there's lots of animals that live on these sponge stalks. It's
Yeah,
just-
zoom out, that's
if we
great.
had a place big enough to put them, I would consider collecting them. It's just they're quite large. You get a lot of biodiversity characterization on one of those sponge stalks.
Bonk. There's another little stalk hiding over here. Oh, sorry.
Getting some Matt Damon quotes in their comment question box now.
Alright. Perfect.
All right, Steve, I want
awesome.
you to, in your best Matt Damon voice.
People, they want to hear it.
I don't know if I'm
It
qualified.
looks like I'm looking at nothing, but here.
There's got to be something about growing potatoes on Mars or something.
Yeah.
Yeah.
This one's from Goodwill Hunting.
Can you do a zoom right where my lasers are? Sir.
I can stop
I don't
bothering
see
you about it,
anything.
Steve. I'm sorry.
Oh yeah, there's
Just
there's
one quote. Just one.
Just one.
There's a small bamboo coral stalk and a
With
small
is that
polyapigon
a
sponge.
Sponge?
Yeah, in the background, and then
Sorry, I
there's
was
a coral
blocking everything. There we go.
very tiny coral branch.
Oh yeah.
Yeah, bamboo coral.
There we go. Finally, I'm sitting on the
Hoping
ground,
to
I
see
think.
some
Whoop.
bamboo corals that were a little bit bigger than 5 or 6 centimeters. It's really tough when you sample bamboo corals so small, sometimes they don't get a chance to branch yet, and it's one of the really important diagnostic characters.
All right, zoom out.
The type of branching.
Just watch out on the port side.
The presence of branching or where they branch, yeah.
Right, I think, was it Fanny that was showing the nodes versus the in-between and how they branch at different spots, maybe?
Thank you for catching that.
Yeah. I guess it's on its starboard side, right? Yeah.
That's the starboard side, yeah.
Yeah.
It took me a second to orient, but yeah. All right, I'm turning back over here.
You want to do another move?
Yeah.
All right.
Yeah,
All right.
let's do another move.
Okay.
220?
Yeah.
Did much of that layback come out or no?
Yeah, we swung.
Yeah. Nice.
I believe we did.
Nav, where are we at waypoint-wise?
We're
I'll get back
almost
over
to
here.
6.
All good, thank you.
Yeah, you can, so that screen on your left is called high pack, and you can see where the boat is, and you can see the WP on your left side.
Yeah, this
That's
thing?
the point.
All right, here I'm just gonna sit down for this one.
Oh,
There you go.
right here, Steve. These
Yeah.
guys. Okay.
Yeah, so the Hercules is the circle with the cross coming out,
Yep.
and then Atalanta is the square.
Gotcha. Yeah.
It used to be a square.
Is that just
No,
a
it's, no,
sponge?
no, it's a yeah, that's
That
square.
one.
Yep.
Yeah.
Is this just a sponge polyp or something?
This is a actually a carnivorous tunicate.
Oh,
Yeah, we saw
okay.
one a bit earlier in the dive.
Yeah. All right, we can zoom out.
Very nice. They're also very difficult to collect. They gellify pretty quickly in surface temperatures.
All right.
For those of you just joining us, we are on our second dive of Expedition NA174 to explore the waters surrounding the Republic of the Marshall Islands, looking for possibly new species or range extensions, other amazing biological discoveries, as well as geological specimens.
There's a sea pen right here in this pool of sediment.
Yeah.
No, it's a coral,
It's like
actually. It looked like it was in the
trio
sediment.
of things.
Yeah.
Do we want to stop the ship here?
Another
No,
sponge
just
behind
a
it
quick
too.
look.
Up on the little ledge, maybe.
Yep.
All right. Which would you like to zoom out
The
first?
bottom center.
Okay.
The crynoid we saw recently, and then the Polyapagon sponge we saw a few times. Yeah, so this is also another bamboo coral unbranched.
Levi, maybe you can
Oh,
control
those
the
are
camera.
shadow.
Yeah.
I thought
Yeah,
that
yeah.
was another branch from
Me-
far away.
You staying right here?
Still
Yeah.
pretty
Turn
small.
left, holding.
Perfect.
I'll tell you what I'm
Hoping
doing.
again
Yeah.
to make a bamboo coral collection here for something a little bit bigger, but these are pretty small. So we'll keep going.
Is that full zoom? All right.
Yep. Nice.
Try to get closer to things next time. Did you want to look
That's
at
okay.
any of the other ones?
That's a good zoom. No, we already imaged the crynoid and the sponge earlier.
Roger. Okay.
Mhm.
Yeah, come on out.
Copy.
Perfect.
Move complete.
We want to let it settle out first before. I don't see anything steep in my
Yeah,
sonar, so.
thanks, Steve. We could call another one in if
Yeah.
you wanted to keep
It
moving.
is look-
Let's do
it
another.
looks like it's
Okay.
flattening out a bit.
Oh my gosh.
What is it?
See something floating?
What is it?
Where?
It's a crynoid. Oh my gosh.
Is it a
this
swimming?
Woo!
Is this your moment?
This is my moment.
This is it.
This is it. She's never seen one.
I've never seen
And
one moving.
So
this
excited.
is Rise
Oh my God,
first.
look at
She's
it.
flying. It's gonna be beautiful.
Wow.
This is a magical moment.
I better
Woo!
not go. All right, we're gonna try to follow this
got
for a
it.
second. Let's.
This is a crynoid on the move.
Can you control my
Beautiful.
pan and tilt?
You want me to tilt stuff?
Yeah, please.
All right. It looks perfect. You're
Yeah,
doing
try
great.
that.
I'm gonna leave it right here.
It's so graceful. So pretty.
Nice shot.
Wow.
Nice shot. The tilt is pretty chunky, so
Yeah, I know.
maybe just fly it with the
Yeah.
verts.
What's that below it?
I think it's a
What-
polychaete.
Oh,
or
polychaete, yeah, that's
something.
what I'm
Oh yeah.
Oh,
What?
This is
That's incredible.
rush hour traffic. Everyone is trying to get to work.
sorry, I'm very excited.
It's amazing. Wow.
Wow.
Oh, he's coming really close to me.
He wants to meet you. Or they want to meet you.
It's, wow.
Oh, it's beautiful. All right, you can zoom out. I think he's pretty close to me now.
Wow.
Oh, he got tired. Now he's drifting down. They get worn out. That's a lot of energy
Yeah.
for them.
Not very, like,
Oh, hello.
you know.
Back at it.
Yeah, video, you just can do whatever you want.
Looks like he's playing,
Yeah,
in,
I'm
the,
just trying to keep up with him.
Yeah, all right.
playing in the prop
That's
wash.
cool.
He's moving pretty quickly.
That's pretty neat. Thanks for indulging me, everybody. And then the little polychaete just like zooming around.
That was great.
Nice shot. That was fun.
Alright, you're moving now. Nice. Cucumber.
It's meant to be. Ryan,
Yeah.
you got your wish.
I can't believe I just said that I hadn't seen one yet, and then
Well, I've never seen a whale. So
yeah, true. Everybody put in their wishes.
I was going to say mermaid.
Oh
but-
yeah. Definitely new species to science.
I've never seen a
let's see
giant
what this is
squid
over
fighting
here.
a sperm whale.
True. What's this? Is this another one of those sponges we were looking at earlier?
This is actually a tunicate. It's a stalked tunicate.
Ah, okay.
Yeah,
Look,
it's
his
a
name is
filter,
Squishy.
filter feeding
Let's take a
organism.
look at it. What's squishy?
There was a jellyfish
yeah,
just went
there's
by.
a
He's really cute.
Tetragaster.
Oh, really?
Yeah,
Okay.
he gone now.
All right.
A tunicate.
Let's look at this tunicate then. All right. Zoom in.
This is a stalked tunicate called Culeolus.
Is it?
It's
Dying?
very much alive, actually.
Okay.
Yeah,
It kind of has a brown hint to
yeah,
it.
you can see
Yeah.
where it takes in water.
It's like a floating paper bag.
So-
They really are not the most graceful or beautiful of animals, but you know,
Steve,
They're
you said it
Oh,
was
he's got a little
Culeolus?
associate
Culeolus. Yeah,
Culeolus.
Stalks
C-U.
there?
Would you add that into the little data logger
Yeah,
real
yeah.
fast? Thank you, thank you.
All right. You can zoom out. Are those little squat lobsters on the stalk, or?
They're probably isopods or
Yeah, okay.
amphipods. Yeah, you find on any vertical structures typically find a lot of
Yeah.
critters,
They still
crustaceans.
kind of stretched out, aren't I? Sorry.
No, you're good. You got tether.
Why am I not?
Let's see. Oh, what was that? What is what's up in the water column. I got anything? There you go. Something to look at.
What is it?
Shrimp?
Oh, shrimp. There's shrimp everywhere. Wonder how many shrimp there are in the ocean.
Oh, wow.
Well, actually,
Alright, you want to zoom in?
if you take an invertebrate zoology class, you'll learn that probably one of the most abundant organisms on Earth are actually copepods.
Wow, look at it. Look
they're
at it
probably
swim.
more abundant than insects.
That's
if you consider
really
the
cool.
depths of the ocean.
Oh, yeah, it's an interesting angle looking down its body like that.
Yeah.
Its eyes really look like
It's
they're
quite
glowing.
far away from you.
Yeah.
This is a swimming shrimp, Penaeid.
Alright.
You wanna do another 20 meters?
Will do.
220? Let's
Yeah.
do that.
Bridge nav.
Oh, I see us.
Oh yeah, can we look
Coral right
at
here?
this?
Yeah.
All right, you're gonna land it before we zoom?
Yeah. Just give me a second, I'll get closer. I think I'm stretching out a bit, but
Should be getting you closer soon.
yeah. But yeah, I'll get a
Kind of reminds me
landed
of
zoom in that one.
Chrysogorgia energy, kind of like fireworks.
There
Yeah.
you go. You guys don't even need me anymore.
Alright. Let's see. I can get closer if this doesn't get us. Alright, go ahead, video.
Oh,
Little polychaete.
something swimming by, yeah.
Yeah, this is Chrysogorgia.
Alright.
And can you tilt down a bit?
Yeah.
Get,
I can. You want me to do tilt?
Yeah,
Do you want to fly?
sure.
There you go.
Yeah, that's full zoom.
Yeah, if you want a closer one, I can get closer to it if you need to.
Do these Chrysogorgia have an associate species as well, Steve?
They do. It has one squat lobster.
Yeah.
What do you think about collecting this? It would have to be like the whole Bottlebrush.
Yeah.
Yeah. You want to stop the ship?
If you can grab it near the
Bridge
base, usually you can get the squat lobster to come with it and stay there.
Is the squat lobster kind of like
It's in the
the
lower
bottom?
third
Yeah,
here,
that's what I thought.
Yeah, alright. Roger.
This one is nice because it's not too big, and it's not, but it has the association.
Right.
They get actually quite large. You
Oh,
get
wow.
sometimes up to a meter. This Bottlebrush shape. And we saw one only on the last dive. We haven't seen any of the Bottlebrushes on this dive, and they're often the hardest to identify taxonomically. The polyps are really small, and you have to often look at them under a microscope to determine species.
And so you said Bottlebrush Chrysogorgia?
Yeah.
Back well.
There's
There
a
we
couple type of morphologies of Chrysogorgia. There's planar, biflabellate, so two planes kind of growing in parallel, and then Bottlebrush.
I'm stable there. I can do.
Are you good for me to use the arm?
Yes. Go for it. Are you okay with that position for cycling?
Yeah, looks
Okay.
great.
Do you want a closer zoom on it, Steve?
Why don't we go ahead and snip it, and we'll zoom in the jaw once it's
Roger.
snipped.
And where are you going to want to put this sample?
I think we should go in with the bryozoan.
Okay.
Yeah. The right side. What's that?
Yeah, do you want to
Omega.
see more of your arm?
Yeah, that'd be good. Oh, you guys don't have a... Oh, you're in continuous mode.
Yeah, sorry. I have a preference for that, but...
Yeah, do you guys not like to double up boxes or...
It sounds like we're gonna go in the forward box again.
Forward box, alright.
Yeah.
Correct?
Yeah, we can double up with the
Nice.
bryozoan in omega.
Yeah. Was that the brittle one?
Yeah.
Right.
Yeah, you want to center it up on the species, on the specimen, and so I can try to see where it's coming from. Is it attached on the bottom or the?
Yeah, it's going to be attached
face
kind of
is
on
it?
the bottom, like probably back here. But there's a crab in the lower half that we really need to get with it. So you might want to like position this way, you know, and kind of get close to the rock.
Like get your jaws behind it, I guess.
Yeah,
Yeah, what if
I
that's,
get
what I'm
like-
trying to say.
Like, kind of get in there.
Oh yeah, looking good.
Yeah. Sometimes it's hard to cut these low when they're like in a.
Sometimes you can also like knock it off the rock,
Yeah.
Oh,
Oh, I think
awesome.
we're gonna get that.
that and then pick it up.
Going.
I think you got it. Yeah. Good, good, good.
Hopefully.
All right. Let's go with that.
I like all the polyps like close up.
Great job.
All right, I'm retracting. Oh, don't let him
And
go.
then the squat lobster is still part of this one sample, right?
Right. Yeah, it's part of the same sample.
All right,
the wrong
so
index.
retracted. You want your forward box open?
Yeah.
Okay, forward box is coming out.
I think that little skull lobster might still be in there.
Yeah, I think so too.
Yep.
There we go. Should be fine there. So you're going into omega?
Omega.
Is yeah.
Yeah.
Let me know if you need any other views or anything, Levi.
I guess it's perfect.
Thank you.
It's okay if a piece falls into lambda. It's not the end of the world.
Okay. That should do it, right?
Yeah. Come on,
Get
little
back
buddy.
in there.
Oh, maybe.
Oh, oh, oh,
Oh, oh,
oh.
oh,
All
he's
right.
going over there.
So, all right.
Okay.
Not the end of the world. So we'll call that one sample. And we'll just
Okay.
make sure it's a
I'm
no.
closing the box now,
Squat
so
lobster
that
went
we
in
have
the lambda, but it belongs to the Chrysogorgia.
Noted.
Okay. So
Beautiful.
you might
Good
not
job.
put anything else in that forward box if you want to keep that little guy specifically. Because it'll probably
Yeah, it was on the move.
it'll probably try to escape after that.
Right. Yeah, they're typically not very active. So we'll see how the rest of the dive goes.
Okay.
They'll oftentimes a lot of crustaceans will find a corner and they'll just sit there.
Okay. Cool.
That's fun. I think that was my first
Yeah.
sample with Herc.
That was awesome.
Oh,
Yeah, that's
first
great.
everything.
All right. We want to keep going. You happy?
Happy.
Okay.
You want to do another 20?
Yeah.
Looks like you've been stopped and you got room to go,
Yeah. Let's do it.
so. Yeah. Yes. This sounds great.
Oh, hello. What's that?
Shrimp or amphipod?
It's a little bit close to me to,
You're about to hit your, yeah, you're good.
yeah,
Zoomed right over that rock.
just barely.
Yeah.
I'm still figuring out if I like this sea bias, but
Yeah, if you're too light, you can
I
dial
think
it.
I think I actually feel like I keep, I
You
keep
sinking?
hitting, what is this? Oh, that's just a rock.
Oh yeah, just like a
It looked like something though.
It did, yeah.
No, I actually feel like I'm having to pull up a lot, but maybe I'm just figuring it out still. I think the terrain is just uneven. I think there's something behind this rock.
Something red, little,
Yeah, a little bit of red. Oh,
Yeah.
I'm pretty stretched out, aren't I? I'm not gonna.
yeah.
Lots of comments, Levi. Great job.
So another shrimp.
Thank you. That was fun.
Another shrimp.
I'm gonna, oh, something bigger. Is that another shrimp? Bonk.
Shrimp are everywhere.
The shrimp.
There's got to be so many shrimp. I think every dive I've been on has had shrimp.
Alright, I'm
Just
gonna
everywhere.
step back because I'm a little stretched out here. Maybe we can see some stuff back here.
Yeah, ship is moving, so we should get you
These
some
little,
Yeah.
shortly.
like, waves on the sediment, is that caused by current?
Yep,
Okay.
probably deep water currents.
Is that anything?
They're in the same orientation as on the previous site, so
Mhm.
again, we expect kind of north-south flow.
Right.
Is that anything in that rock there?
I don't see it.
No, I think it's just sand playing optical illusions.
What'll really blow your mind is that the sand that we see here is actually the dead shells of millions and billions and trillions of marine plankton.
Oh, wow.
Yeah, none of this is sand like that you would
What?
find at a beach.
Really?
Bonk.
Yep.
Oh, 'cause
So
it's
sand that you find at a beach is probably eroded. Could be coral rubble, you know, calcium carbonate. It could be eroded rock. It could be anything. It could be, you know, like broken, like sea glass. You
Right,
know, stuff erodes over time. But all the sediment here,
Is that
since
a
we're so far
shelf
from
or what?
land,
Has to be
Looks
things that have
like
died
it,
and
yeah.
fallen.
Atlanta
Yeah.
can see over it, so,
so
it's
there's
not
a
super
there's
big.
Yeah.
a group of organisms called foraminifera,
Should be interesting though.
Mm-hmm.
Looks like
and
there's stuff on it.
foraminifera form shells that are kind of like tiny balls.
Either it's sand
And
or
those
something.
shells sink over
Yeah.
time, and it forms this
Those
kind
look
of
like they're gonna be corals. Though,
uh,
maybe?
you know, very fine but porous sediment.
Are they like a diatom or?
Kind of. There are diatoms that are spherical, but these are different. They're single-celled.
Maybe
They
that's
have
just sand.
shells, and their actual organism is more amoeba-like,
Okay.
if you can imagine that, but with a shell.
Okay.
Are you talking about forams?
Yeah.
Are those the ones that are in the water column that have like, it's like a ball with little spikes coming out of them?
Yep. Yeah. Those.
They're really small.
Some are
Video,
called.
do you wanna
Yeah.
zoom
Those are
just
called
past them in
radiolarians, but it's a similar type of plankton. Yep.
That's super interesting.
No, it's all good.
But you'll see when we
didn't
take up
notice
the
it until
push
now,
core we
so.
took earlier, we'll have some excess sediment that we can take a look at,
Can I tilt you up a little bit?
and you'll see what I mean.
Oh yeah.
When it dries,
I just
it's
wanna look at this
smooth
stuff.
like
yeah.
pearls.
Oh,
yeah.
wow.
They're pretty tilted up already.
Yeah, I'm
but-
excited
I'll try to
to
lose
yeah.
that
see the
mount.
samples when
Yeah.
we
Yeah.
bring them back up.
There you go.
There you go.
We'll probably be working through the night, so
Happy to help.
Staining
Yeah.
or-
There's something really interesting on this rock. It's like,
May have to
Can't
take a
tell if
video
it's-
of you
there's
for my
quite a bit
feed.
here. Yeah.
yeah.
We've got sponges and
Alright, what do you think? I
corals.
think. I don't know if we'll drift any more.
Yeah, that sediment looks so smooth.
Levi, what do you think? You think do a small move a little bit closer?
No, I think you're good.
Okay.
I think you can approach this
'Kay.
and then just look at it while Atalanta swings
While we
in.
sit out, yeah.
Yeah.
Yeah,
Yeah,
okay.
yeah.
Start circling things.
Yeah. Let's move up a little bit. There's
Yeah, you
some
want to look
stuff
at
to-
the interesting,
to the top left.
what this,
Oh, it's another
yeah.
hole in a rock.
Yeah,
I love it.
Oh.
just watching my upper
Your brow's
here.
pretty close, yeah.
Yeah,
So this
I'm just going to get a view of kind of what I'm doing with here. Okay, yeah, lots
yeah,
of cool
let's
stuff.
take a look at both of these things. So there's one
Here,
thing
look at
and
that.
thing two. Thing
Thing
one,
one
thing
and
two.
thing
Oh,
Was that from?
alright,
Was that
do you
Alice in
wanna
Wonderland
zoom
that
on
had that?
the zoanthids
Thing
Dr.
one-
Seuss?
first?
Dr. Seuss.
Yeah.
Okay, I couldn't remember.
Okay, go ahead. And I'll kind of pan around.
Oh, sea spider.
Oh yeah,
Oh, picnigonid.
awesome. We had an interaction earlier today with an aquarium, and one of
Yeah,
the
so this sea
kids
spider's
asked
actually
about sea
eating-
spiders.
This sea spider is actually predating upon these
I haven't
zoantherians.
tried your auto XY
Wow.
yet.
Oh,
You
yeah.
could try auto XY and see if it just
Yeah.
see if it just floats there.
It's small, so it's going to be hard to focus on it, but
I
they
kind of want to be a
eat,
little bit closer, though, before I hear him.
they eat those.
Here, can you, video, can you zoom out?
You
Because I
said
think
they,
we're going to spend
what
a
are
bit
they
of time
eating
here.
again? I'm sorry.
Zoantherians,
gonna
it's
get
like a
set up
colon.
properly.
Think of it like a colonial anemone.
Okay.
Yeah.
I did that thing where I was like worried my head was gonna hit the, and then I was pulling back, but like that. All right, let's do shift over a little bit. Oh, not bonk. All right, do auto XY.
Oh.
Yeah,
I see something.
Okay. Let's try that.
I see something.
Zoom in. Is my head right on that rock there? I think it was. Bonk. Sorry, I'm coping with this overhang bit. I'm relearning autos, try not to destroy cameras.
Yeah, you might just float it.
Alright.
Overhangs are tricky.
Yeah.
It's pretty small, so I think in the interest of time,
Yeah.
I would actually rather like to set up for a collection of this if you can.
Yeah.
So this was the thing that was in the background in the rock
Okay.
that we couldn't sample before,
Oh, wow.
and
This is gonna be an
this is a
interesting sample.
Chrysogorgia, what we saw earlier. We probably collected,
I
but
see
this
two
is a
options.
planar
I see
species.
land on the ground.
But
I
this
reach
one,
up,
I know
or
we're
you float it and I reach in and grab it and then we float away.
Let's do a Niskin bottle here too.
Okay.
I'm gonna start Niskin and then
So much activity. It's so exciting.
Yes, another, I think these overhangs and crevices are
Yeah. Just full of life.
creating heterogeneity for
Totally.
organisms. Yep.
so
Yeah.
So
Let's try to float it. I think you'll be too high if I sit on the ground.
let me know if we're setting up for the grab or the Niskin first.
Yeah, you wanna do Niskin first? Or you wanna do coral first?
Let's do the grab first. I'm
Copy.
floating here.
Coral first.
Okay. I'm gonna pause the pumps 10 and 11 right now. Okay, since we'll be here a bit. So pausing 10 and 11. Thank you.
Confirm, thank you.
Yeah, I'm not sure I can get much closer. Here, I'm a little bit worried.
Quick question.
I would say grab what you can, and if it ends up grabbing a lot, that's okay. It's pretty small. So
Yeah.
grab what you can from it. It's gonna be on the fly, right?
Yeah.
It's gonna be on the fly. Auto XYs didn't
And do that. Do the trick for it.
dropped me into the things. Okay. Yeah, you want to bring your arm out and then let me know if you want me to pan your cameras around or anything.
See, it's going that
Do
way,
we
so
know
maybe-
where we're thinking about putting this? We have corals in both of the forward
Yeah.
bio boxes.
I was
Probably in starboard.
I was
box.
thinking about putting it on top of the rock in the starboard
Cool.
outer.
Yeah, we've got rocks in both of them, so either,
Okay.
or two of
This is a
them.
good angle for me. Or do you want me to nudge arm in better?
And then, Steve, do we have any
You wanna go
guess
grab
of
it?
what this is
Yeah,
beyond
go grab
just
it.
coral?
I'm super stable.
Yeah,
For some reason.
let's stow it
I think it might be too far
first.
away.
I'll put it in the chat.
Sounds good, thank you.
Okay, let's
I
see
think
if
it's too far away.
I'm gonna come up very slightly and nudge in a little bit. Just
Or instant sea log.
be ready to.
Yeah, you move whatever you need.
I'm just gonna do this really slow.
Yeah. Do you have your brow against the rock or something?
I think so.
What you got going on? Nice.
There we go. Alright. I'm gonna auto head this and not hit that. Not a fan of that. Okay. You want me to come down a bit?
Yeah, you gotta come down, yeah.
Okay.
Easy.
And I can.
You're pretty close to
I
it.
know. I'm coming up.
Come back a little bit. That's good.
Okay.
Alright. Can we try to just grab a piece?
Yeah, just do it.
Alright.
Come on.
Get in there. Yeah. Nice.
Great job.
Nice.
Great grab.
Okay, I'm backing off this wall.
Yeah.
Just let me get out of here and then you can.
Yeah, where do you want us to do it?
Probably a starboard box still, or did you want to stick it on top of the corals you already have in the front box?
Whatever Steve wants.
It's going to be pretty sinky, so I think the starboard box in one of the outer bins,
Right
we'll just
here?
throw it
Okay.
in there.
I'm gonna sit on the ground,
Okay.
just so we're
Stable.
stable. That's all. As long as you're all right with that.
Yeah.
All right. Auto XY and
That was a tricky one. That was like under and underhang.
I know, that was a
Right.
great grab.
Yeah, very good.
I don't know if you know this. I have a
Alright.
reputation of choosing very, very difficult collections to make, and it's not my fault.
Okay.
It's the
I'm
coral's
gonna
fault.
bring up your camera for
It just likes to grow in difficult places.
your starboard box. You can start coming around if you feel like it. And I can pan over as you come around. And then whenever you want, I can switch to a sample salvo that'll give you a better view of the box.
Yeah, so you guys have a big blind spot.
Yeah, you ready for sample salvo?
Yeah.
Okay. So that's just a bigger view. Yeah, so you can see your height there.
I think I'm like pretty.
I think you're a little bit low.
Yeah. You want to draw or
Yeah, you want me
open
to
that-
bring the
open
drawer
that
open?
puppy out?
Okay. I'm gonna turn on PRV. Okay. Starboard box coming out. Slow. Oh.
Alright, which position do we want this to go in?
Let's just throw it on top of that rock in the outer
Is that E?
E. Yeah.
Okay.
Oh, that's an easy one. Alright. I like that. Alright.
I think you're in it.
That'll work, right?
Yeah. So nice.
Good.
Good job.
Super.
Nav, could I get a pin dropped for 055?
Okay, bringing the box closed.
Thank you. Say again? Oh, okay, awesome, thank you.
All right.
Is there a limit to how many samples we can get per dive, or is it just based on like the number of boxes and not wanting to mix things?
No, there's no limit.
Okay.
Yeah, this is with this. We
Turn
tried not to
that-
mix up samples of the same, genus or maybe family, depending on what, the kind of expertise we have on board, because we can discern things
Wait, what was that?
a lot.
Oh, so like
we can
you can
distinguish two
fully
closely or
customize.
similarly related coral genera from another. It helps to keep things separate,
Right.
especially the one aspect which we don't have the specificity for specific sampling tools for is if we wanted to look at small crustaceans that are often associated with certain corals, for example, they attach, and sometimes if they're present in the environment, sometimes they're in the
You got some
box,
tether if you wanted
when you
to
open
go
the box,
up and
the
over
things
that
swim in.
lip,
It's hard
you
to
know?
tell if it came from that coral or not, but it's usually
Gotcha.
Are we still in sample mode
rare,
or
it's
should
the
we switch
minority
to,
of samples where that happens.
custom?
There are tools, or there are sample deposit tools called quivers, which is like the push core quivers on the right hand side,
You
but
really got to
with a
bring the
giant
arm around.
rubber plug on top.
That's pretty-
Sometimes
Yeah, that's pretty
ROVs
fun.
will use those. They'll take the plug off, put the sample in, put the plug on top, so then that sample is
Gotcha.
isolated.
Okay, that makes sense.
Yeah.
I thought we were gonna do the Niskin.
Oh, that's right.
Niskin.
They wanted a Niskin. I forgot about the Niskin. All right, so we've done 3 of them, I believe.
That's right.
Yes.
So probably gonna go. I totally forgot about it. Is your camera retracted? All right, so we've done 3. So I'm gonna look for the 4th one. And we believe that. Alright, so it's number 1, which I think the 4 is right here. Right. I think it's that one. That one.
I don't see the bottle camera. Is it up somewhere?
There it goes. Alright, so it should be the 4th one. Should be.
Yeah, it looks closed. Yep. Okay. Good capture.
Sorry, I wasn't talking about
Yes,
this
it
wheel.
is.
But yeah, 4th one got triggered.
Yeah, this will be 056.
Yeah. Sorry, panning around.
Cool.
Everybody happy?
Yeah, can we just take a look, actually,
Yeah.
just at that on our way out?
Wait, sorry, was the
Absolutely.
Niskin fired?
Let me just refine where my camera is. And then we can...
ROV?
Sorry?
Did the Niskin get fired?
Yes, it
Okay,
did.
sorry, I missed that.
Nope, that's all good. All right.
Great job, ROV, on
Alright,
collection and
it's
piloting.
done.
Thank you.
Okay,
Nice flying, Rye.
thanks. Alright, good arming. That was a challenging sample. Okay, what am I looking at?
So we're just going to do a zoom on this.
Oh, this nice one?
Yeah.
Yeah, okay. I'll get up and...
Whichever one is the most difficult to see.
Yeah, I know, I'm like, oh, I'm
It
already...
looks like there's something else over to the
already
left as
nosed
well.
in.
No, I promise I won't sample this.
It's hard to get light on it, isn't it?
Just give me a sec.
But don't tempt me.
Yeah, try that. And you might have to iris up to get the sample nice, but...
So I just wanted to look at this. So this is the same genus as the coral, the bottlebrush we collected, but it's probably a different species.
This is pretty pink little...
And
bulb
okay, there's a
thing.
couple things going on.
Try that.
Oh my gosh.
Whoa, what's
There's a
those
couple
yellow
things going on here.
on it?
So that's an Aplacopher, and it's a selanagaster worm, mollusc.
Coming down there.
The
It
yellow?
looks like a worm, yeah. So this is an animal, it's a predator of corals.
Oh, wow.
And so you can see the golden color of the skeleton is all denuded of polyps. That's all
It's been eaten.
prey. Yeah, it's been eaten.
What kind of worm did you say that was?
It's a mollusk.
A mollusk.
So it's not a worm per se. It's worm shaped, but it's a selanagaster
All right,
worm.
there we go.
All right,
And which type of coral did you say this was?
Chrysogorgia.
Thank you.
Yep. And it's also got a Europtychus squat lobster.
And I'm panning down there. All right, there we go. All right.
So this thing, I have actually a couple slides on it. Maybe if we have some downtime, I can talk about selanagasters and predation on corals. But they're vicious. If you
Oh,
can
yeah,
tell.
It looks like it has much larger appendages than the other one.
It's actually just one long tube, and they just wrap around the thing.
The squat lobster?
Oh yeah, the squat lobsters have long arms. Yeah. Actually, there might be a couple of selanagasters there, but yeah, all of this denuded branches. This is all predation from this mollusk.
Matt,
All right, thanks for that zoom. I think we can move on.
All
Do
right.
what?
Zoom
Our
out.
different attempts at selanagaster.
Yeah,
Right.
I'll put it in there.
I put a little question mark next to it, like, I think this is how it's spelled. I'm
All
not
right,
sure.
Are we ready to go up and over this thing?
There, so I
All
put
right,
it in
since
the
we're moving on, I'm
Nice.
going to
Thank you.
restart the pumps,
Roger.
10 and 11. They'll be the same pumps as earlier.
Noted, thank you.
Okay? Thank
I'm
you.
Coming up.
Great.
Nice.
That's hilarious, Leela. Neither of us got it right.
Nope.
Yeah? Yeah, let me just... I guess I'm not in a bad spot.
Invertebrate diversity is very cool.
Mm-hmm.
Yeah. I would be good
Super
with
interesting.
a ship move.
I'm a little biased, though.
How are you feeling?
Yeah, if you keep moving. Yeah.
Okay, let's do it.
Yeah.
I think so, yeah, that looks where...
Yeah.
Tilt up a little bit. I keep like craning my neck, like that'll change the camera angle, but...
Okay,
Right now, I
ship
keep
is
looking
moving.
at the big screen, but you could just look at this one too.
Yeah, that's true.
Yeah. Yeah.
Yeah, you're like, what's over there?
Someday it'll be augmented VR style, super wide angle cameras.
I wonder if anyone's gone from the video seat to the ROV seat. I'm always curious, like, sometimes when people start
Big
in one
perspective
seat,
to have.
Yeah.
Yeah.
Yeah. I know Megan...
Or vice versa.
Megan Putz is she's not video but nav, but she's really good. Like her zooms are crazy because she's always so focused on the biology stuff. She's always interesting. Like
Plethora of knowledge.
yeah, when you get cross training or whatever, people pick up interesting skills. But
Mm-hmm.
She's probably listening. Hi, Megan.
hi, Megan.
Hey.
Hello.
We saw a pink coral.
Should probably tell us like 2 or 3 species that it would have been, you know.
Yeah. And a crunchy one.
Slowly working our way up the slope at 24.18 meters.
Is that something in the corner there?
Oh yeah, under the ledge.
Yeah. Oh yeah, that's another
Let's take a look.
Chrysogorgia.
I might have to sit to see that one.
I think a snap zoom is enough. It's looks like a Chrysogorgia similar to the species we just saw. And there's definitely a lot of preference at this site for overhangs.
You want to zoom in?
Yes.
Let's,
Lots of stuff hanging out
Yeah.
underneath these little overhangs.
That tells me that the tops of some of these
pan over.
structures maybe don't have great flow conditions, but when you have a
Sorry, I
constriction
slipped
here,
down this
like
one.
when water has to
You're
flow
alright.
underneath an
There we go.
overhang,
It can
it
speed
Can you
speeds
tilt
up,
my
so
camera
it's
a bit?
Yeah. What do you
better
mean?
for
Just up.
suspension feeding animals.
Yeah,
Nice.
that's
Up is
great.
pretty fast, isn't it?
Yeah.
Very great observation.
Did you cool it off a little bit?
I was trying to, but I might have actually just sped it up.
Can I do it on here or not
Yeah.
so much?
Let's try the PRV. Oh no, you can't on that computer
one's-
because it's down right now. Yeah.
Right.
So tilt
So-
up.
Looks good.
You want less?
Yeah, because tilts up is hot.
Or is this... I wonder if this only changes the these buttons.
There are some
Alright.
small
Or
amphipods
does
on
anything to do with
this.
You want
Some
zoom
small
out?
crustaceans. They live on the
Oh,
coral.
sorry, Steve. I didn't realize you're talking about it.
No, that's good. We got all we need from that zoom.
Great.
Ready to continue.
Yeah, let's see. I mean, it's pretty hot right now in general.
Lots
I
of
think
marine
maybe what we actually
snow.
want to do is we want to turn tilt down up, grab
Make-
her,
make
and
the-
then nothing happens.
Yeah. Oh, yeah. That's because we were fighting each other.
Oh, I see. So tilt up, tilt down.
Down's pretty slow.
It's a little slow.
Up is about right.
Alright.
Going down, going down, going down.
Some
going
of that
down,
might be
going down,
coming
going
from the
down.
vehicle.
Okay, I'm off.
Oh, okay. Stirring up the sediment a little bit.
Yeah.
Yeah.
ROV, when you're driving Hercules and the other ROVs, are you using like multiple camera angles or sensors?
Oh yeah, I mean, I'm looking at how many cameras right now, which Levi's got more cameras on his other ROV, but
Wow.
The third-person view is a real blessing.
yeah.
Some
Okay.
vehicles aren't two-body. This one's a two-body, so you get that third-person view, which gives you some context,
Totally.
which is real nice. Because if you're just
Yeah.
flying by this, you don't know that there's a big rock to your left or
Right,
right. So
right.
you kind of have your third-person view over here, and then you have your primary, what everybody's looking at over here. And then you have some wide angles to kind of let you know where you are. We like to look at the bottom of the ROV because we
Yeah.
like to set it on the ground gently. So we're
Gotcha.
looking at the porch landing, and you guys can keep your nice clean view on your main camera. So we're looking at the wide angle stuff. Yeah.
Gotta incorporate lots of things. Sounds
Yeah.
difficult.
Yeah, and we got cameras looking backwards and at both sides, and then we also have a few that we can switch over to our bio box views and the sample
Right,
jar views. So that's
right.
like when we know a sample has been collected in the sample jar, it's because we have a view of the sample jar.
Gotcha.
Yeah. And yeah, lots of like trying to get spatial awareness from a bunch of like flat information, which
Yeah, for
can
manipulation.
be a little bit tricky. Yeah, sometimes
you
you'll
need at
see
least
us
two
like,
views. Yeah.
sometimes you'll see us like bump something with the manipulator before we grab it because we're
Okay.
just trying to find it in space, basically.
Yeah,
Depth perception's
Yeah.
hard.
It's really hard.
definitely. I could see how it would be very difficult to figure out.
I don't know if I ever realized how much I use my hands when I'm talking because I just take my hands off the stick to talk, but is this a little stalk of something?
Kind of looks like
Gonna
something
Yeah.
there, almost
Zoom
in the
there,
middle.
video?
Copy.
See if, is it just a dead stalk?
Dead stock. Looking
Yeah,
like it.
little.
Something on
There's
the
a crynoid.
side.
Oh.
Yeah,
Is it a
crynoid.
little
Little
crynoid?
Yeah.
Baby crynoid looks like.
Oh,
Little micro crynoid.
alright, you can zoom out. Alright, I'll keep going.
Almost, yep.
It's like a cool design from the lava right there.
Yeah, it's a
Yeah.
cool formation.
It's really
wall.
neat.
It's neat. I like this flat lily pad rock.
Yeah.
It looks like a cartoon dinosaur foot. Alright.
You want 20 more meters? All right.
Yeah,
All
let's do it.
Copy. In which heading?
I think 220 is still good for me.
Copy.
Yeah. Slope might be doing like closer to 250 soon.
Yeah, you want to turn starboard left to the next move just a little
Yeah, maybe.
bit? Okay, all right.
But
Ship
it's
is moving.
so slight.
Copy. Ship is moving.
Alright, I'm just gonna kind of weave back and forth, look for stuff.
ROV, do you know of any ROVs that utilize like VR headsets?
I've heard of it.
Okay.
I think Ambari did some work with it.
Yeah.
I heard a rumor about it. I haven't seen it firsthand.
That would make
Yeah,
a
you
really
can
cool
do it.
video game.
Yeah,
Yeah.
I've heard about it.
A lot of people have been talking about some video game Subaquatica on the
Standing
comments.
in this hole?
Subnautica?
Subnautica, yes. Sorry.
I've watched people play it. It's a really cool game.
Yeah,
I do like a
we'll
little
have to check it
snap
out
zoom in
after
there? I
this.
just
The
wanna see
comments
what's in
have
there.
seemed really interesting.
Yeah.
I think it's just
I play Subnautica.
Yeah?
Yeah?
The soundtrack is awesome.
Okay, cool.
When I'm out on the deck, I just put a little
I think it's
soundtrack
just
in
a
the background.
dead sponge.
Yeah,
Awesome.
dead sponge.
It's my
Yep.
nerd
All right.
in me.
Alright,
Pan,
zoom
zoom
out.
out.
We're getting desperate. We might have to look at the sea cucumber. Oh, I got a little stalk over there. It's a little bit in the weeds. Sorry.
Survivor of a spaceship. Crash on an alien oceanic planet. Sounds fun.
Right? Yeah, I was reading about it too.
Yeah.
Are we talking under the ledge there?
Yeah.
Good if I snap zoom?
Yeah. Let's do it. What's that? Anything?
Looks like an old
Just
sponge stock.
dead. All right. Zoom
Okay.
out. And I'll get back in frame here. Sorry.
A little off to my starboard. I can just turn right if you like.
Sure.
Let me just come to stop at 10 degrees.
I'll get back from anyway. Yeah, I know some of the, there's only a couple ROV specific institutions in North America, but
Okay.
but I know there's one on the east coast of Canada that they at least have like a simulation for ROV piloting. So the people that go through the program there do a bunch of like simulation piloting. I've heard they like to play this game where they try to get a hold of each other's tethers.
Oh no.
Like that, it's like battle bots, but like ROV, which sounds super fun. I wish I got to do that.
I'd watch a show about that for sure.
Is this a sea cucumber? It's slightly more purple.
Yeah, we've seen one like that. We can.
Roger.
Yeah.
ROV simulation. That's pretty cool.
But yeah, I met some of
much.
the pilots that came out of that program who got straight on an ROV and were like pretty good with it. So it seems like a fun way to learn how to pilot.
I'm sure they'll be coming out with new VR training.
I'm sure.
I mean, they do that for the aviation side as well now.
Yeah. I got a little ledge over here.
Yeah, there's a ton of flight simulators.
Yeah, a lot.
But probably not a lot of ROV simulators. ROV's pretty simple though. Like you just need like a screen. You don't need like the plane simulators. They're like pitch and roll, and ROV's pretty simple.
I was really scared when I started like piloting her because I was like, I don't game, and I was like a lot of the people I know are gamers who pilot, and I was like, I'm gonna be so bad at this, like, but it's nothing like.
Your lack of gamer expertise would really set you back.
I think spatial awareness is actually probably your biggest skill, like just understanding where you are in space relative to your equipment. But
I know, I'm always really impressed the way, like, y'all can set down the ROV just, like, on the seafloor. I don't know.
I'm very, I don't have a good sense of left and right. Like, I gotta make the L with my finger.
Me too, every time.
Yeah, and so you get in the seat and it's like, you know, you're using compass directions and left and right and port and starboard and clockwise, counterclockwise, and yeah, just like getting your head around all that was. I prefer if people say starboard and port. I still don't. Left and right doesn't make any sense, but. Well.
Copy.
The way I remember starboard and port is port has the same number of letters as left.
But the problem is if you don't know what direction left is to start with. Yeah.
Also, update on the Subnautica. They have a new version, Subnautica Below Zero.
Oh, we got a stock over there.
There's a new one coming out as well.
There's a big one in the distance on the right.
Yeah, I'm
on the
totally gonna have to
starboard,
get this.
Working towards that.
That's a pretty big looking stock.
Yeah. Probably be a little too stretched out if I get there yet, but.
It's another dead Bolosoma. Bolosoma with the.
You want to do 20 more meters,
Right?
right?
Yeah.
Okay.
Sorry, I haven't been keeping on top of.
Just same direction.
I think so, yeah.
Yeah.
220. It's good.
Do you want to look at any of these small stalks here?
It
Those look.
not really. I'd rather
Okay.
keep moving.
Roger. I am a little bit waiting just for Atalanta to catch up with me,
Yep.
so
Okay.
this is a good chance to just look at frivolous.
Ship is moving.
We'll let you know if I see anything.
Yeah.
Interestingly, all the Bolosoma sponges we saw on the mega outcrop a bit earlier that had the corals and the crevices had 0 ophiuroids on them, but only the dead stalks
Is a black
had
coral
the ophiuroids on it.
Really? Oh, wrong.
Oh, you mean down
See
here?
what I'm looking at? Yeah.
Yep. Well, let's take a look at it.
Just getting over. Yeah. All right. Get a little bit closer to it. Video, you want to go for zoom?
Go for zoom.
Pan over.
Yeah, this is another.
Great spot.
Yep. So that
Oh, sorry. I don't know why I'm... There we go.
this is heteropathes. We collected this earlier in the dive.
Is that not... So that's not a black coral?
It is a black coral. Yep.
I'm learning.
Yeah, you can kind of see
Yeah.
that black on the bottom there.
Yep. You can see through the tissue.
Cool.
And another trick is if it ends in pathes, it's a black coral.
Good
Because
to know. You said it was called heteropathes?
this is a heteropathes, yeah.
All right, you can zoom out.
So we're gonna move on our multipuffer. We'll stop pump 10 and 11 now,
All right.
and start pumps 12 and 13.
All right. Let me get
Thank
those
you.
plugged in.
I'll get a feel for that vignette one day.
Oh, it's all good. I mean, most people do zoom out till they see it and then zoom slightly back
Yeah.
in, so I wouldn't expect you to be able to find it without seeing it.
Data, are those sample numbers?
Those will be samples 057 and 058.
Thank you.
Anything else? Oh, I got a shrimp.
So I've been
And
looking at the.
Those will, sorry, those
No,
will
no,
be
go-
You
pumps
want to do a
12
quick
and
zoom
13.
on that, just because we're
Yes.
Thank you.
Oh yeah.
The shrimp? Sure.
just get in there, see if I can follow him for a sec.
I love those swimming shrimp.
Nice.
They're so neat.
I do like their little glowing eyes.
Mm-hmm.
All right, you can zoom out. I'll get back to the seafloor. How are gauges and stuff looking?
Gauges on Herc look good.
It was flattening out a bit, which might explain why some of the biology has gone away, but it should pick up a bit. Between waypoint 6 and 7, we're going to enter a steeper slope.
Sounds good.
Ship movement complete.
Yeah, we can do another one. I don't see anything coming up in the sonar, so.
Do you want me to do a dive log?
Yeah.
Okay.
I feel like you know where to find everything.
That's
All right, we're
fine.
going to do another ship movement then.
Yeah.
So I'm going to stop and restart pump 13. We had some issues with that one yesterday, and it seems like we might be having issues with it today as well. So
Okay.
please don't record it as a sample yet. We'll restart that one as a sample after I...
I have unfortunately already recorded it.
Okay,
as a
well,
sample.
have to, I guess, update it after. I've just turned it off, but I'm going to try restarting it.
All right, I'll note that down.
Samantha.
Ship is moving.
Roger. There's a dead stalk there.
So you're looking at thrust, that's the comp. It's hard to read that.
Yeah, so I just know
Annette,
from memory that's
do you
six,
want us to delete
six,
pump 13?
I don't know yet. I'm troubleshooting.
Okay, okay.
Thank you. Yeah.
Data, can you hear me? Cool. Hold off for a moment, Annette's gonna see if she can get it working. All right, thank you, Data.
Huge anemone right there.
Oh yeah.
sorry, I was off SBL for a sec. Let's go look at this. Let's get a nice... I'm just trying to practice setting down here, so...
Nice.
Give me a sec. All right, you want to go for zoom?
Go for zoom.
This one's up a bit. Good. There we go.
And then, Annette, whenever you have a free moment, I'm ready for the volumes for 10 and
Nice
11,
picture.
but no rush if you're troubleshooting.
Nice. Oh.
Okay, just bear with me. Thanks.
Not
All
a
right.
lot of current.
Good image, and we can move on from that.
Roger.
What is this?
Anemone.
That.
For now.
Is there any other thing on this rock you want to look at?
No, not particularly.
Alright. Yeah, there's another one of those
Yeah,
tunicates.
another tunicate.
Beauty. What are you mostly looking for, Steve? Hey, sorry.
Anything and everything,
Yeah.
but mostly new things we see
Yeah.
that are different from things we've seen on this dive or other dives right now.
Roger.
So I'm not interested in getting a lot of repeat images,
Yeah,
but
cool.
if there are better looking representatives, maybe.
Yeah.
But just trying to get a diversity, estimation of the diversity here.
Sounds good.
Ship movement complete.
I think, yeah, this is again, it's kind of a flatter, low rugosity type of environment.
Let's do another ship move.
So when we get into the slope, it should change a bit.
Copy.
Yeah. I'm still happy with it. And that's still taking us towards the next waypoint? Yeah, okay. Nice.
We're passed by waypoint 6 and headed to waypoint 7.
Look at this little, like, there's like little pebbles now.
Those look like
Oh wow!
nodules almost.
Yeah, little.
Look like nodules, crusty nodules.
Yeah.
We just got a question about how we plan our dives and how we map these, asking if we use LiDAR or some sort of laser scanners. So we actually use a multi-beam sonar system. So basically we send down acoustic signals to the seafloor, they bounce back, and they give us kind of an overall picture of what the seafloor looks like. So similar to on land, We have lots of topographic features, valleys, ridges, mountains, open plains, and that helps us to basically pinpoint where we want to do our dives. So thank you for that question, also from Fayetteville, Arkansas.
Looks like we got another ridge coming up with maybe a
I think my
stalk on
friends
it.
may be watching.
Yeah, nice. All right. It's very tall.
Okay, I'm ready to give you the numbers, but before I do, I just wanted to tell you, I've got pump 12 running as expected. Pump 13, I'm getting some confusing readout here. It seems like it is communicating and the time is on, but the flow is not being recorded, so I don't know if it is pump is actually the pump may be on,
Dramatic
or
pan up there.
and it's just not being reported, or the pump may not be on. So I'm going to leave it on, assume just in case the pump is actually working, and we'll have to figure it out later. So you may want to make a note of that.
All right.
Or we'll circle
so
back to it afterwards.
Can we take
I'll make
look
a note
at
of
the
that,
Yeah.
and then we'll
Okay.
just keep the sample for the
Sorry,
time
it's just
being
adjusting.
in
Yeah.
the hopes that something's being
yeah,
She's
let's
captured.
pretty
Yeah.
loud in that ear.
All right. Okay.
And then
Yeah.
I'm ready for the volumes
Great.
whenever you are.
Sure. Okay. So.
I'm just gonna get
Just
close
find
to
it
this video,
again.
and then we can kind of do a zoom where I'll pan up the.
So pump 10,
Absolutely. Yep.
Yeah.
262-157.
Yeah.
Cool.
You can zoom in,
262-157.
and I'll kind of go
Yes.
up the stalk.
And then pump
Mark this
11
down to that.
is
Yeah,
156-815.
156-815.
Yeah, so pump 10 had a very high flow rate,
It did. Yeah.
anomalously high.
Nice move.
So we'll have to see.
Steve, is this a
All
bamboo
right.
coral?
Thank you very much.
Yes.
Okay,
Thought so.
thank you.
Do you want to look at any particular part of this?
No, that's okay
Okay.
for
Okay,
now.
zoom back out.
Roger.
Ship movement complete.
Yeah. All right. I mean, this is a pretty shallow ledge. I would be fine if we kept moving if our goal is to kind of truck along.
Copy.
Did you want to look at any of these corals over here? This was like the one we sampled, wasn't it?
Which one are you pointing out?
I'm looking at, it's kind of hard to see. It's right above the crynoid. Do I have my lasers on here?
Oh,
yeah.
it's really, really tiny?
Yeah, it's quite small.
Oh, yeah, I see it now.
Okay.
No, yeah, it's okay.
Now we can do the next move at a bearing of
Okay.
230.
Actually, I changed my mind.
230.
Can we do a little haircut on this coral? Take a piece off the top.
Yep.
Yeah, don't call any moves right now if we're gonna do sample. Yeah.
Ship's holding.
Yeah.
Alright,
Nice little haircut.
This,
So we want a haircut on this
I heard
tall
about
bamboo?
someone
yeah,
Yeah.
let's see
just a little off the top.
if we can get like 15 centimeters of it,
Yeah.
maybe 20 centimeters.
Okay, let me see if I can get any of my autos to hold here.
Where are we gonna put it?
Yeah. You want to see your thing more?
So
There you go.
This I think we can also put it in the same place we just put that white coral in the starboard box with the rock in it.
Awesome. So that'll be starboard biobox E.
Yeah. It's pretty sinky, so it'll float down. I think you're gonna want probably half.
Are you happy with where I am here?
Yeah, you want me to grab it?
Yeah.
This is when you test your depth perception. It's like you
Yeah.
don't know if you're gonna be in front of it or behind it.
That looks
Is
pretty.
that enough, Steve?
Yeah, that's good right there. Super.
Beautiful.
Nice.
Alright. And this is a starboard box
Yep.
situation? Okay, let me get your camera.
This will be 05, yeah,
Nice.
059.
We'll
Yeah.
flip
59.
over as
Making
you
a
go.
target.
This is a different
Thank you very
unbranched bamboo whip than the one we sampled yesterday.
Oh, wow.
The polyp morphology on this one is much more The polyps stand much more erect in that they probably have sclerites that are inhibiting them from
when you want to add
collapsing,
a sample salvo.
retracting fully.
Yeah, you can switch it.
Okay. And I'm gonna bring you up a
Very,
little bit.
very interesting.
So with this height, how old would this coral be?
Very difficult to tell. In most cases where age dating has been done
Ready for the
successfully,
box?
Yeah.
you would
Alright,
need to
strawberry
grab
box
the
coming
base, the
open.
very part of the rock where it's oldest and attaches to the rock, or the very part of the coral where it attaches to the rock.
Where in the box are we putting this?
Biobox E.
E. Okay.
Should just stuff right in
Just
there.
giving you easy stuff.
That's a good warm
We'll save the inner boxes for rocks first, and
Okay.
we'll put the biology on later. But since we've already got a rock in E, we can load it up with some bio.
Alright, that looks good for me. I'm gonna bring that closed.
Can we zoom on the remnants before
job,
we
Arlo.
Yeah.
take
close
off?
here if
Yeah,
you wanted
thank
to
you.
get out of there.
Yeah.
I think he said he wanted to zoom in on the remnants.
Yep. Okay, I'm gonna come back out of autos. Get the arm out of the... Alright, video.
All right.
Are you wanting to look at the top of it mostly?
Yeah, top of it.
Okay.
Yep.
Alright, arm is secure. It should be pretty close
Zoom
to
in
this
there.
stay
and I'll-
position.
As tight as you can get it.
Yeah.
Do you want me to change the salvo or not while you're moving?
I
Alright.
just leaved it for now.
Oh. Yeah, so you can actually see the sclerites of this coral. They kind of look like tiny little needles in the side of the body wall of the polyp. That's what's providing the polyp some rigidity. And those are also taxonomically
Sorry, I
informative
lost it
characters, so we'll be looking at those in the lab when we get the material back up
there. I
on
think
deck
my porch might
Very
to
be
determine
under the
the
ledge
genus
a
and
little bit there.
species.
Okay, I'll
Awesome.
just zoom
Okay,
back out.
zoom out,
I
please.
think that's okay.
Are they used for protection too?
Okay,
Possibly.
let's get resituated.
They
'cause
can.
you're-
They definitely have very sharp, pointy morphology for some species.
Okay.
But strangely enough, it doesn't really protect them against their main predator, which are sea stars.
Oh.
Am I good to move on?
I think so.
I'm gonna come along here and get
Yeah,
back
sea
into you,
stars
and
will predate upon bamboo corals regardless of how spiny they look. Mostly because they're really fleshy. They have a lot of tissue, so they're very nutritious
Gotcha.
for predators.
This is
And
19.
were you telling me that
Can
bamboo
we go
corals,
back
I
to
may be
dive
mistaken,
custom
were these the
salvo?
ones that have a
Yeah,
lot of
absolutely.
mucus as well?
Yep.
Okay.
Yeah, there's
Yeah.
very common in environmental DNA.
We'll hold off on the movement.
samples.
Just a
Just wanna see
second.
what the...
Just have her look up a little bit. Yeah,
Yeah,
one-
thanks.
just a moment.
Just wanna see what's gonna crop up here. Alright, going over this.
Okay.
And then I'll get kind of out in front of you.
So, what have we learned this watch in our last 15 minutes? Anyone want to summarize?
Make sure your auto depth is actually on.
Yeah, those buttons kind of like click but don't go sometimes.
Heterogeneity in the
You get enough
environment
green is
can
good.
lead to
Yeah.
more
Well, sometimes they just
diverse
don't go on,
species.
so-
Yeah.
Lots of corals are hanging out in overhangs,
All
likely
right.
because there's a better flow-through rate
You want to
there.
do a move?
Yeah.
Yeah.
All right,
I'm
let's do it now.
feeling good.
230. Yeah.
And our ROV pilots are really good at collecting
I
samples.
had the sampling expertise was really good this watch. A lot of really round, nodule-y looking rocks in this area. I'm hoping as the slope's about to pick up, probably when the next watch comes on, But yeah, we have a little bit of different types of habitat here,
Look at all these
different
round rocks.
slopes.
I know.
It's like a bunch of turds.
It's just kind of like all of it seems like it all just all of
Scientific
a sudden
term,
happened.
of course.
Doesn't it?
Exactly.
Doesn't it look like a bunch of round?
Funny.
Yeah, like
funny.
a
Giant deer ones.
yeah.
Is
Something.
this a
Ooh.
coral that we've seen before?
Another
Steve?
Chrysogorgia.
Yeah, we did
Yeah.
sample. This is one of the
Okay.
Bottlebrush Chrysogorgia. Yeah, we did sample that earlier.
I know there's like 12 million different kinds of pink coral, so
Oh yeah, yeah, pink. Yeah, pink's a very common color in the deep ocean. Actually, that was a question you asked earlier, and I didn't answer it because something got distracted me.
of why?
A lot of pigments. So the thing about evolution is when you gain a trait, it's very hard to lose it.
Ah.
And so I think a lot of the
what's this?
color, especially the warmer colors,
See this little fish
are
video?
ancestral traits that are just
gonna try to
retained
look at
by
that.
deep water species when they
Gotcha.
radiate in the deep ocean.
When they
Another rat
had
tail?
them, maybe when they lived in shallower areas.
Is that a small rat
Yeah,
tail?
is
So
there a
that-
reason?
That looks very similar to the one we slurped earlier. Yeah, it looks that's a
Yeah.
cusk eel actually.
Oh, okay.
It's
Yeah.
I should know
it's
this.
an eel.
Drazen would be
That's a
very
cusk
mad at me.
eel, yeah.
Cusk
Oh.
eel. Very cool.
Cool.
Mhm.
All right.
Cool.
Cusk.
Pulling up.
But it could also be something related to their diet. You know,
I'm
if
just gonna
certain
check
corals
my-
maybe
might-
consume preferentially, but
might be
it's very unlikely. It's probably
not
more of
centered on this.
an ancestral trait.
Okay.
And is there a
Yeah,
reason
that
that
would
pink
explain
would have been an advantageous color
some things.
in ancestral populations?
Well, if you think about it, a lot of the you know,
I was like, I feel like
it's
I'm driving.
widely
I just
considered
keep
that
driving
a lot of the
crooked.
coral diversity in the deep ocean probably radiated from the shallow into the deep ocean, and so they would carry with them traits, you know, of their
Look at
shallow
how
water
long this-
ancestry. However,
Oh,
I'm
look
not
at that
sure
old
about
dead
deep
Stop.
water
Stalk
is,
radiation
yeah.
in primarily
like
deep
a
water
meter.
families. There's a lot of deep water families that have very bright, pigmented colonies. Like the primnoids I'm thinking
really
of
long.
mostly
Is that another
are
stalk
thought to
on the
have
ground?
radiated from areas around Antarctica in the
Yeah.
past. And sometimes you get very bright
Good look.
orange, very bright red, or even green
You can look at that.
corals
Green. That's cool.
in the
You going
areas
for
where
that
we're
stalk?
in.
Yeah.
It's either very
All right.
pale white,
See if there's
translucent.
anything living on it.
All right.
Not
zooming in.
particularly.
I'm just kind of waiting for Atalanta, so we're just finding things to look
Steve,
at.
we just got a question about how we process samples when we bring them back up. So we do have a small lab aboard EV Nautilus, but maybe you could tell us a little bit more about what we do
A
there.
little polychaete, maybe?
Oh yeah, maybe right next to it. Yeah.
Yeah.
All right.
Yeah,
zoom back
every
out.
sample that comes on
Alright.
board will get imaged and water samples will get filtered. We preserve everything, usually multiple different ways, so it's useful to the
There's something. What's in that sonar to the port side? See that? What's the range
yeah,
on that
that's
ring?
a pretty big something.
Holy cow, that's 60 meters away. That was a serious range on that unit. Should we go over there? That looks cool. Should we turn?
Where are we looking?
Yeah,
You can chase down that target. Yeah.
that's what my gut says is something's over there. So you want me to turn? We're gonna turn port and line up for that.
Maybe it's that whale or giant squid we've never seen.
Mm. It's a very
Yeah, I bet
very
it's a giant
still.
Still fingers
It's
crossed
a
for that
very
mermaid.
still thing.
High likelihood.
So this, whatever this is, it's probably a good 10 meters tall. It's pretty high shadow it casts.
12.
Wait, so which screen are we seeing?
still
again?
don't see
So
what
you can,
we're looking at.
So we can select that head, and then we can increase the speed at which
The sonar
because right now
is
gonna
over
take
there,
forever
and there's
to
two different
rescan it.
sonar heads. The one on the left belongs to Hercules, and the one on the
Yeah.
right belongs to
So, like,
Atalanta.
Okay.
Select,
And
select,
it sends out
select.
sound, and it bounces back
Right.
Alright, and
reflective
then I can
things.
do
Each
scan
one of those
speed,
rings
plot.
represents a distance.
Okay.
Do those
And so by
range,
counting how
minus
many
range
rings out
plus
Right.
and multiplying by
scan
the
speed plus
distance
speed.
per ring, you
Okay.
get an idea of how far that
There
thing
you go.
is out.
Now it's gonna
Gotcha.
that'll ring
Makes
it a couple
sense.
times.
And things that are higher profile,
Right.
larger, will scatter that
There you go.
sound
Okay.
Lobby.
more
Alright,
than
so I gotta
something
go
that
look a
is
little more port.
smaller.
All right. Learn something new every day. Learn lots of new things
Alright,
today.
I'm at 200 right now.
Is that
I'm gonna
another
go to
shrimp
one.
on that rock? Yep.
I'm looking 190 now.
Oh, jeez.
Here we go, let this scan, and we'll see how close it is to my bow here. Should move a little bit to the right. That's, oh, it's a little bit dimmer
Shrimps
now.
are omnipresent on seamounts, but they're very difficult to collect. Even when you try and suction them, they swim out of the way pretty quickly.
All right, I'm gonna go 10 more degrees to port, and then we'll go that direction. All right, 180 is what I got. We'll scan it and see it. So I'm looking 180, and yeah. See if it's close. It's kind of weird. Huh. Yeah, we kind of turned away from, we're kind of cutting the slope. Yeah, 180 is like towards that target. Yeah, you want to go 20 meters closer to it, right? All right. Yeah, 180, yeah. Still kind of uphill.
All right, we're gonna get a little closer to this target.
Yep.
And then if it doesn't pan out to be anything, next
They can
next
just
watch
turn.
will get us kind of back on.
Yeah.
All right, that's fine.
Just see if there's anything more interesting.
It was ringing pretty good when it was.
It was, yeah.
Maybe I adjusted the settings wrong or something. I accidentally pressed range minus. I don't know if that changes the. Is it like an auto sensitivity or is it like a? I haven't played with it yet.
I don't know.
A lot of times there could be a gain button. Range, speed.
Sorry, just kind of waiting for you. So,
Yeah.
looking around.
Trying to think of what jars are available. I don't remember, to be honest.
I think we have 3 jars left.
Three
Yeah.
jars are.
There is only, there's 7 and 6. The
Oh,
one
2.
labeled
Okay.
flush we can't use,
Yeah.
right? So yeah, just 6 and 7 left.
Okay.
Yeah.
Flush is the emergency jar. If we find something that's just like we have to have it, we can throw something in
Yeah.
the flush at the end.
But it'll likely have a lot of detritus from other flushes, like
It's
from other
alright.
areas and
Yeah,
stuff. So.
okay. Noted.
Yeah, because like we'll use that jar anytime we use a suction sample, but don't actually take, like we don't actually get it into the, oh, hello,
Oh yeah, another
into the jar.
flytrap
Whoa.
anemone.
Yeah.
No.
Wow.
Anemone.
All I think of is,
Oh,
I think it was Finding Nemo where the
Oh.
kid couldn't say the word.
just dragging
anemone.
myself along.
So when you hit the ground,
It's
you
more
don't
common
expect
than
it.
you
It's like
think.
Yeah.
Oh, I guess I am 0.6 meters. It's
Hello.
probably because I'm looking way out.
Hello.
Hello, I'm anemone.
So something to note is that these anemones typically orient facing down current,
Oh.
and so they're really good bioindicators of what the flow conditions are like at that site, because they don't move very much.
Yeah. Okay.
Yeah,
That's awesome.
Since we've seen them, primarily they've been oriented downslope,
Uh-huh.
so you know you would
Makes
expect
sense,
water
it's
to flow
coming-
downslope.
Yep.
But at this site, it's more variable, so it's probably also why we're not seeing a tremendous amount of suspension feeders in this spot. It might be squirrely currents.
I think video's doing handover, so we'll just kind of, and we've seen a few
Yep.
of these now, right?
Yeah.
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 So we'll just kind of
Hang out.
hang out. We're getting pretty close to watch change here, so I'll just keep taking looks. at
Yeah, we're
things.
gonna transition to next watch and back row now.
All right. Let's see. Hello. Hello, Isabella. My friend.
My friend, it's good to see you in the
It's good
in
to
the
see
herc
you.
in the herc seat.
Thanks.
What a nice surprise.
All right. Well, hello, goodbye. Yeah. All right.
I'm good. Appreciate you.
Mic check.
Hearing you loud and clear.
All right, got you. Yep. Nev, you got me?
I got you.
All right. Video, you good?
So where are we headed?
So it seems like we were heading kind of southward. They were picking up on like a potential wall, kind of hiding out maybe over here. And so they wanted to start taking some steps this way. But also in a general, we're going to, maybe after we explore a potential wall, we'll keep going up slope.
Okay. So we're gonna head south. Is the ship moving right now, or?
No, I let that goal kind of
Okay.
sit down for a second. But if we're ready to get going, I can keep the ship movement.
Yeah, let's.
Fanny, are you happy to keep seeing.
Yeah, so we just want to continue going upslope. I think they're coming up on some kind of wall ahead of us, so we can go check this out and just keep going upslope.
Okay,
Sounds
Roger. Okay.
Quite a bit heavier than yesterday.
All right.
Okay. Well, if anybody is joining us live on our website, nautiluslive.org, thank you so much for exploring with us today. This is the 4 to 8 crew once again. So this is Brittany speaking. I am the Science Communication Fellow for this watch, and we're super excited to be back. We started off this dive early this morning, and we have now gone through 12 hours of our dive, so we are back on the 4 to 8 watch. So we are continuing to explore the seamount. Our main objectives are to look for what are the biological as well as geological components of this particular area. So the seamount has never been explored before, and it's really special that we're able to have the opportunity today. So just kind of looking around and seeing what we can find. And once again, if you have any questions or comments that you would like to submit to us, you are certainly able to do that if you go to our website, nautiluslive.org. We do have a little chat box there you can submit, and we'll do our best to get to your questions and comments.
Oh, there is a ledge.
Yeah. Danny, are you fine with me turning off your aft light?
Yeah, go ahead.
Thank you. You're starting to get to the end of your tether.
Yep. Just got my leash pulled.
Yeah, if we can, I think it would be cool to check out that ledge, see if there's anything hanging on it.
Yeah, I just have to wait for Atalanta to catch up.
All right.
Is the ledge the 10-meter thing, or is that something else?
Possibly.
Okay.
We'll see very soon.
I know, I'm like...
Suspense,
the
right?
anticipation, I'm like, what is that?
Exciting,
That's
yeah.
10 meters. Yeah, I wanna know. Someone's asking what would we like to see this shift. I want to see it all. See everything we can. Yeah, I don't have anything in particular. Just personally, I'm here for the exploration, seeing what mysteries are down here, what kinds of things can we explore or uncover. I mean, I do like biology, so anything that is moving around, doing something, I really enjoy those. But honestly, I'm here for all of it.
That's crazy.
That's pretty impressive.
It looks like it's circular, yeah?
Yeah.
That's crazy.
Amazing.
Is there almost like a cave down below? No.
We're not going in a cave. Don't worry.
Oh, no.
Hey, Danny, just to give you a heads up on your craft arm. as far as position goes. It's a little
Such
bit out.
interesting features.
Yeah.